So, thank you everybody for coming, and on behalf of Helsinki Python Meetups, welcome to the first post-COVID Helsinki Python Meetup. I counted you quickly, and there's roughly 50 people in here, so I'm actually feeling a little bit agoraphobic. I'm not sure if I've seen this many people in a year and a half, and I'm really glad to see all of you here. I'm also glad to be here myself. And hopefully we can continue having these meetups roughly every other month from now on for a long time without any further interruptions. We're here in the Compi Center, hosted by Qvik, and we have two excellent speakers, Jerry and Juha, who will be talking to us about two quite different kinds of topics today. But before we get to our speakers, a quick word from our sponsors.
Hello again, my name is Jari Sandström and I'm from Qvik, and I work here as a technical recruiter, so welcome on my behalf also. And if you have any career related questions, feel free to ask me because we are hiring. Yeah, thank you for all of you for coming, and let's get on with the show.
Right, so something weird happened to my resolution. This seems to be like 8K monitor or something. Right, hello everyone, let's get started. So my name is Jerry, and today I'll try to convince you all to start using typeheads. If you're already using typeheads, that's great. Hopefully you will still learn something new from this presentation. So I work at Vault as a competence lead of Python. We have around 100 people who are actively using Python. It's not only like the data language, it's also the biggest backend or web backend language at Vault. So I would say that we have quite a strong internal Python community. Before we get started, let's try to get some motivation. So how many of you have seen these kinds of exceptions in your Python applications? If someone is not raising their hand, I bet they are lying. But yeah, the basic idea is that you expect something to be something or out of some type, and then it's out of some different type, and then world crashes. Maybe an even more common one is that something is none when you expect it to be some proper type, and then you try to do something with that, and then an exception is raised because none type doesn't do pretty much anything. So wild none type appears. It of course depends on the application and the frequency of these exceptions, like how bad they are for the business or whatever you are building, but in my experience, these are quite bad, and especially they are very it's something that you're doing wrong in your code when you see something like this. I have a simple solution for you. Start using typeheads. that will basically enable you to catch those type errors or potential type errors already offline, maybe on your own computer or latest at the continuous integration phase. But most importantly, before you deploy something and then they happen in the live environment. As you may know, it's not enough to just use the type hints. You also need to check whether they are correct or not. Here I list a couple of those static type checkers. MyPy is maybe the de facto one, at least in my opinion. Then there are a couple of alternatives. But yeah, throughout the presentation when I talk about static type checker, I'm mainly referring to MyPy because that's my weapon of choice. When you combine the type hints with the static type checker, you get something like this. So in the above or the upper visualization, there's some simple code, some very dummy code, and in the lower visualization, yes, much better. There's the MyPy run over that same code. So basically, if we, for example, look at the second line of the MyPy output, argument foo to my function has incompatible type string expected int. So basically, that's string, sorry, while the expected is int. And similarly for the lines 10 and 11. Okay, so that's simple. Just introduce the type hints and start using the type checker. Are we done here? Sadly not. How about all the great dependencies that you have? And luckily, the Python ecosystem is very rich, so you don't need to reinvent the wheel. Every time you're implementing something cool and new, there's a high chance that there's already some ready-made mature package for that. But is it typed? Does it have type hints? If it's popular and well maintained, there's a high chance that it has the type hints baked in. Just a couple of examples, like Flask just recently added type hints to their source code, like did PyTest like a year ago. So I would say in general, the more popular the package is, and if it's maintained or not, depends on whether there will be type hints at some point. Of course, if you're active member of the community and it's super important package for you, maybe consider contributing the types to the upstream project. And even if there are not types baked inside the dependency repository, there's a high chance that there are type stubs. For example, in the typeset repository, it looks like this. It's underneath the Python organization in the GitHub. And if I open the stubs, well, it's very small. But maybe the most important bit here is that you can see that there are plenty of those. And these are not from the standard library. These are all third-party libraries. So for example, you may have heard about requests. It's here. And yeah, mainly the popular ones. Then if you cannot find it from there, and if you're brave enough, you can have a look at the directly from PyPI. If you search for types, you get more than 10,000 results, but I would be careful with this. Those are not necessarily what you're looking for. MyPy is also often helpful. So for example, if this requests example, if you're trying to import something from there, MyPy is kindly suggesting that maybe you should install the stubs from that. I think it was in the latest major release of MyPy that they stopped bundling the type or the type shed stubs to the MyPy package. So now you need to install those separately. But MyPy is very helpful with that. All right, let's say you're very unlucky. You couldn't find it from the typeset either. That's not a problem. You can create the stubs yourself. And there's even a tool for that, which comes with the MyPy. And usually you're only using one or two functions of a dependency that you're using. So you don't need to add type hints to the full-blown library. It's usually just those two functions that you're interested about. But yeah, if you're heavily using that library X and you build stubs for that, please consider open sourcing them, because I bet there are others who would be very pleased to have those. In case these stubs are something new for you, just briefly, let's consider that we have some dependency, and this would be the source code file for that. It's a very dummy one. Then when you run it through the stub gen, you get something like this. So it's PYI file. And this is a dummy type stubs file. You can see a couple of any's, and that's pretty much it. And the purpose of this stub gen is that you get kind of a dummy template that you can then start filling with the type hints. And like I mentioned, usually you're only using a couple of functions or a couple of types from the library that you're using. So it's not like you generate 20 modules for some library and then fill in the types for all, but rather for the ones that you're using. That's already very helpful. So here in the lowest screenshot, you can see an example where the types would have been filled manually. So although the stub gen is not kind of giving you the correct types, it's giving you the template, like fill in here. So that's already really helpful, at least in my opinion. All right, now we have kind of the tools for adding type hints to your project. How about if you have like a massive existing project? Is it too late to add type hints? Well, at least MyPy supports gradual typing, so you can start very small. No need to create a massive change set where you introduce everything on one go. And we actually did this kind of exercise at Vault. We started around 2 years ago, and one example project, our biggest one, was at the time over 100,000 lines of code. And the main learning from this exercise was that when we started using types, we wanted to gently enforce them for all the new code, and then kind of be looser for the let's say, legacy stuff. Like, in other words, strict configuration by default lose when needed. And if you're interested about this topic, I recently wrote a blog post about it, which you can have a look. I'll share the slides after the presentation. But yeah, for strict by default lose when needed, as a practical example, it looks something like this. So in the upper part of the, let's say this would be like mypy init file, for example. You have relatively strict configuration. By default, mypy is quite loose. So if you install mypy and run it over your code base, if you don't have any types, it will kind of be okay with that and exit with 0 status code. And of course, you don't want that if you want to have the types. So you need to turn some knobs. So by default, very strict, and then separately for each module that you had, have it, for example, to disallow untyped defs. It's like double negative, but maybe you get the idea. No need to have types is basically the translation for this line. And then this is actually like very fun Friday afternoon activity to get rid of these legacy module special cases. Or then when you're adding something new to, let's say, this my package legacy module, I think it's a good practice to start by adding the types and only after that adding the new feature or like the change that you are doing there. And with this strategy, you eventually end up with fully typed project. Of course, it takes time. It depends on how big of the project is and how active you are during the Friday afternoons. Then I bet someone in the audience is thinking like, okay, I have a very mature project and I have 100% test coverage. Should I still kind of consider doing this? Yeah, I think you should. There was very timely blog post from the folks of urllib3. Just, I think it was last week. And well, the main gist was that they added types. They discovered tens of bugs. They are the second most downloaded package in PyPI, and they had 100% test coverage already before that. So I bet your project is not as mature or as well tested or has as many users as urllib3. I think this is a great motivation to start adding type hints. But unfortunately, this is not enough. Like static type checking is not enough, at least in most cases. So you always need some runtime type checking as well. What I mean by this, let's say you have like piece of Python code. It can be a web app, some data pipeline thing, whatever. Almost always you need to mangle some data which is from some untrusted source. Even if it's from the database, it's usually considered untrusted source. Especially if you have like write permission to that database, then you can go and change the data and it becomes untrusted source. So type hints plus static type checking can handle and help you here, but it doesn't help you on the edges. As a practical example of this, no, we won't go to practical example just yet, but luckily there are tools nowadays which help you with the runtime type safe things. Hopefully most of you have heard about Pydantic if you have been using Fast API, which is maybe not the newest and coolest framework out there, but becoming the most popular one. They're using Pydantic quite heavily, but it's not the only use case for Pydantic, so I think there are plenty of use cases for that. The basic idea is that it utilizes the type hints and checks them during the runtime that they are correct. So for example, in this uppermost example, it will verify that 1 is int and false is boolean. But be aware that Pydantic does some magic as well. So if it's not the type that it will expect, it will try to convert that. So this is also okay for that. I think for the boolean, it accepts 0 and 1 from the integers. I think there are some knobs in the configuration of Pydantic if you want to be more strict. But most often, the being a bit clever is actually helpful. But you just have to be aware of that. And here in the lower examples, it will raise Pydantic validation error during runtime. And yeah, I mentioned practical example, so consider that you have some code which fetches some data from some API, and then you expect the shape of the data be something like this. So you just create an instance of my API response with the data that you got from the API. The difference between the red one and the green one is only the import. So the data class import in the upper one is from the standard library, while the one in the lower one is from Pydantic. And the difference during runtime is that the lower one will check that the types are correct, or will try to convert to the type that you have. If it fails, it will raise. While in the upper one, you can very easily have like foo, which is boolean, and your code will crash at some point. So this is very like practical example of the edge.
Marshmallow data classes also does the same thing.
Yes, they do. If there's something that you maybe should take home from this presentation, I think if you remember this visualization, it will be enough. So this is the same image that I had earlier, but with these green things here. So runtime type checking on the edges. For example, Pydantic can help there. And then the type hints plus static type checking for the actual code. And same goes for your dependencies. Don't forget those. Don't ignore those. For example, if you get some data from here and you think it's integer, and then you pass it along deeper inside your logic, but it's happened to be string, it will crash at some point. And it can be really deep in your logic already. It may be even like security risk in some cases. But something to note from this image as well. So there's a performance penalty in doing the runtime type checking, which is why you should only do it on the edges. So if you're using Pydantic models, for example, deep inside your logic, I think by default it's doing the runtime type checking. That's like running your application like handbrake on. So MyPy can handle everything else except the untrusted data. So do the runtime type checking only on the edges. Somebody not convinced yet? There are some added benefits in this. So not only you can catch the potential runtime exceptions already when developing locally, but you also get superpowers in your IDEs. And I argue that there are less testing needs, less documentation needs. For example, you won't need any tests which have a line assert is instance something. That's already covered by the static type checker. And I guarantee that if there are new people joining in the project, it will be a lot easier for them. That's basically everything I have to say about this. Hopefully you are convinced now. If you're not, happy to discuss further. Do you have any questions?
Can the Pydantic do kind of like if your, for example, if your API responses do change and you get a new fields, obviously that is a kind of like on general level that's type change, but if it's in certain cases that could be like okay because it's some extra field. You're just interested in certain fields because your application might not need those. Can it handle those or is it like
So for the online audience, the question was that can Pydantic handle the extra fields basically, right? And yes, it can. I can't remember whether it's the default behavior or you need to turn some knob, but at least you can turn that knob per model. So yeah, that's totally possible. Yes.
Are you using MyPy because it's the original, or have you compared alternative software?
Yes, good question. So the question was, am I using MyPy because it was the original one, or have I compared? Actually, for me personally and at my workplace, MyPy has served so well that there hasn't been a need to let's say benchmark the other ones. And for me personally, at least, it's a valuable thing that repository lives underneath the Python organization in GitHub, whereas the other ones were Microsoft, Facebook, Google kind of things. And then I think there are, Guido himself has been behind MyPy. Of course, it has some roots in Dropbox. And then there's some other core developer also participating in the MyPy development. So I would personally consider it as the de facto static type checker at the moment. But feel free to disagree, of course.
A follow-up. Can you speculate why Facebook, Microsoft, and Google all made their own?
Yeah, good question. So the question was why do we have so many of those? As a speculation, I've been thinking about this, and I bet there are some opinions in the internet, but I think these have been developed in parallel, at least partly. So everybody had the idea and then were not willing to give up at any point. But that's just speculation. Go ahead.
Are you familiar with pipeline?
Typecard. Yes.
Okay. You didn't mention it in the runtime type checker.
Yeah, I didn't mention that. Yeah, that would be like the native solution indeed. And there are plenty of libraries as well, like for example the bear type is one of those. But yeah, type card is one of the native ones. But I don't think it's like if you have a big JSON blob and you can model that as a simple data class kind of thing. Type card would be probably very heavy heavy implementation to achieve the same.
Well, we can discuss that later.
Yeah, of course.
Are you also familiar with monkey type?
Which one?
Monkey type.
Monkey type.
Yeah, I don't remember if it's made by Dropbox or Facebook or what, some big company anyway. It basically has you running the program and it fills in the types automatically.
Oh, yeah. Yeah.
Blockchain, but it makes educated guesses
Yeah.
by looking at the actual type being passed.
But yeah, so the question or the comment was about monkey type, if that was the name. I think it works such that you have a, for example, test suite.
Yes.
And while you run that, if you have 100% coverage, it can fill in the types. Yeah, indeed. I didn't mention that. Have a look at that if you feel like not manually doing things. All right, I think let's leave the floor for the next one, and yeah, happy to discuss any Python or typing related things after the presentation. Thanks.
Great. Thanks, Jerry. That was a really nice talk. I learned a lot about type safety in Python. And thanks for the invitation to have me here. This is my first time in a Python meetup. I think this talk will be quite different from many of the talks usually given here, but I think and hope that many of you will find this interesting. So I'll talk about my area of expertise, which is computational genetics, and I'll talk a little bit about the computational aspects of this kind of work in the end. And first I will talk about what we have done during the pandemic. So my background is quite computational, and I've learned the genetics and biology in the last 10 years that I've been working in the field. So first, as context, some of the things we try to accomplish in human genetics research by studying genes and how they affect diseases, for example, finding new genes that affect whether you will get type 2 diabetes, by that we can then further learn about biology and these diseases themselves. And pharmaceutical companies are nowadays very interested in genetics research in general, because this research can help them in developing new drugs. Here I've mentioned an example of a finding that was made in research in 2003. It was found that there's a certain gene called PCSK9, and in people who have a very overactive version of this gene, they can have very high cholesterol levels. And then after a decade or 15 years, then there was a drug on the market that exactly inhibit this gene from doing its work, so that people would have lower cholesterol levels. And in the future, we also hope to better prevent disease. This will be an ongoing work of decades, of course. But one example I want to mention here, we recently found a genetic mutation that is very specific to Finnish people. We barely observe this specific version of the gene anywhere else in the world. But then if you're a woman and you happen to have this version and mutation of the gene, you will have a very high risk of getting breast cancer. So then the question is that should the women who are carrying this version of the gene, should they be screened for breast cancer more often? And this raises a lot of ethical questions, and not the topic of this talk, but these kinds of questions are there already. Okay, I'll now talk you towards a certain finding we made in our research related to the ongoing pandemic. And in order to do so, I'll first walk you through some concepts of genetics. Many of you may remember these from high school, but let's go through them. So in our bodies, we have trillions and trillions of cells. And in each of those cells, there's a set of chromosomes that contain our DNA. So in each of our or most of our cells, we have 46 chromosomes. 23 come from our father and 23 from our mother. And then our DNA is very tightly packed inside those chromosomes, and our genes are certain segments of that DNA. And so every cell, despite cells are very different in your eye or in your liver or wherever, in every cell, basically the DNA is the same, except for cancer cells, for example. And DNA is also a series of these nucleotides, and there are 4 kinds of them in the DNA. They're called adenine, cytosine, guanine, or thymine. So we talk about A's and C's and G's and T's. And one special chromosome is the X chromosome, because females have 2 copies of the X chromosome and males have only 1, because then they have the Y chromosome. And now I'll walk you through a basic concept of an analysis that we often do. So suppose here on the upper row, these are imaginary individuals who have contracted the SARS-CoV-2 virus. And then on the bottom, there are just individuals taken from the population. Maybe they haven't had the virus. And now, suppose we are thinking about that X chromosome, and we are looking at one specific location, one nucleotide along that chromosome. And because women have 2 copies of the X chromosome, there are 2 nucleotides under each woman here, and because males only have 1 X chromosome, there's just 1 nucleotide. And if we now look at the composition of C's and T's between these 2 groups, we can see that there's 1, 2, 3, 4, 5, 6 T's in the upper group among the people who had the virus, and then only 2 T's among the people who maybe didn't have the virus. So then we could think that maybe this specific T nucleotide in this specific location of your DNA has something to do with whether you get the virus or not. Are you following me this far? Yeah. Excellent. So, because this leads us to this, and this is an analysis that I run last June. And on the previous slide, there was a toy example. We don't typically look at 6 people against 6 people, but here we have compared more than 100,000 individuals who have had the virus, and we have compared them to more than 2 million people that we call population controls. So these are just the general population. And these data come from around the world. I'll talk more in a bit. So then I'll explain this figure. Here on the x-axis, you see those 23 chromosomes running. On the x-axis, there are these chromosomes, and on the y-axis is the so-called statistical significance of a specific location in our DNA. So then, in these kinds of figures, we then see these peaks, and these peaks mean that in those locations in our genome, there is a statistically significant difference between people who had the virus compared to people who didn't have the virus. So, for example, there's a very strong, statistically strong hit here on, in the beginning of chromosome 3, and this tends to be different if you have had the virus or if you didn't. Now, I have to mention that this is not a black and white thing at all. This may be like 1%, 5% difference among the two populations. But now, thinking a little bit back to the previous slide with those people with those X chromosomes, I want to highlight this one location here on the X chromosome. So most of the people have only the T nucleotides in that specific location. But then 0.3% of people have a C nucleotide there. Again, because females have 2 X chromosomes, they typically have 1 T and 1 C, and then males just have C because they have 1 X chromosome. Now, turns out that someone who has this C nucleotide here is, in this analysis, this number can change a little bit in the future, but this is the ballpark, would be 30% less likely to get a coronavirus infection. So just one specific location in your genome, in your X chromosome, if you happen to be among those 0.3% of people, you would be a little bit protected from the infection. And sometimes when we make these findings, we don't immediately see any biological meaning there. We thought there's somewhere in the genome, but what does the gene do? We don't know. But this specific finding happens to be just next to a gene that is called ACE2, and this gene creates a protein which is active in your lungs, and this is a receptor for the coronavirus. So the coronavirus binds to this specific protein made by this specific gene in order to enter your body. Are you still following me? Great. So now you understand this concept of genome-wide association analysis. That is one thing we continuously do in human genetics. So we look across our genomes. Is there a difference between people who have a certain disease or have gotten a COVID infection? Now I'll talk a little bit of the background of this analysis. So we established the COVID-19 host genetics initiative, host genetics as in human genetics. And this is a global research initiative that has grown very quickly. So this was launched in spring last year, and this is led by Andrea Ganna and Mark Daly, and then I'm leading the analysis. And this initiative, we generate and we share and we analyze this data related to coronavirus infection and hospitalization. And when I say that it's a truly global initiative, this is quite unique actually in human genetics research nowadays. Because often we have samples in our genetics research projects from Europe, from the US, but not so much from other parts of the world. But here we have now had samples from Korea, Thailand, Japan. And in our latest analysis, so we have repeated this analysis in the past 1.5 years. The latest analysis is from June. That's the result I just showed you. And in that analysis, we actually included 61 different studies from about 20 countries. So our next data freeze, so then we will repeat this analysis, we will find new genes. That's actually this week. So depending on how the data looks like in the next weeks or months, we will have a new result. The first article was about this was published in July. There's always a delay in scientific publications, so when you submit something for publication, it's not there next week. So the analysis results that are in this article, they are actually from January. They are not the newest ones. But then we are going to update this article as we get new analysis results in. And when we do get those new analysis results, we immediately make them available on our website. So that's what I wanted to tell you about this COVID research that we've been doing. Now, for the remaining of my talk, I'll talk about the computational aspects of how we do this research. Because we have all these A's and T's and C's and G's, and they come from the blood samples of people, but eventually they are bits on our computers. So these are the tools or languages that I most often use. And when I say that I'm a scientist, I do genetics, people say, oh, you must use a lot of R. But in fact, I don't. I find R very good for statistics and making figures when your data set is quite small, but it was never designed for the large-scale data sets that we nowadays have. Then we also use a lot of Python. I use Python maybe even more than any other language. And I use that for writing different kinds of scripts that we have to write one of quite often, and different analysis statistics, and then also web server side stuff. Then some of our analyses are not that so computationally intensive, so we can just write it in Python and we don't have to really care how it exactly works inside the computer. It's fast enough. But then sometimes we do have this more computationally intensive analysis, and then we use C++ for those. And then I use a lot of bash and just awk scripts because I often get these text files. They are a little bit messed up. I need to fix something about them, transform them, filter them. And awk is an awesome tool for that. Still in human genetics in this year, it's really, really good tool.
It's better than Excel.
For sure. And then I've also used a lot of JavaScript. I'm now learning TypeScript finally in our results browsers and visualizations that we then make of our analysis in the web. And then some of our computation, it requires quite a lot of compute resources. So then we have this is developed at my workplace in Boston, WDL. It's a workflow definition language that you can use to spread your compute in the cloud. Okay, so this is a Python meetup, so I'm showing a bit of Python code here. This is actually or a piece of code that generated those analysis results I showed in the other figure earlier on in the talk. So, and it's quite I would say, typical kind of code for us. So we calculate some statistics for certain things, and that's pretty much all we do. We often use SciPy and Pandas, and then Hail is a library that is specifically designed for genetic datasets. Then last thing I want to mention, because often people ask me about this, like how big is your data and how is the computation. So here is an example, and this is not related to the COVID stuff I was talking about earlier, but this is about the FinGen study that I'm also much involved with here in Helsinki. So in our current FinGen, FinGen overall, it's a multi-year, it's a 6-year project that is a collaboration between all the biobanks in Finland. And we are doing the kind of analysis, for example, that I showed in order to find new genes that affect different diseases. Not just COVID, but asthma, type 2 diabetes, rare diseases, everything. Then this project is funded largely by pharmaceutical companies, so they can also make their own analysis on the data. And currently we have more than 300,000 Finnish individuals in the dataset, and hopefully in 1.5 years it will be 500,000. So that's 10% of the Finnish population, so maybe some of you are actually in this data. And then for each individual we have measurements of more than 20 million genetic variants. I was talking about nucleotides, but this is actually variant, but I made things a bit simpler. And then we now have more than 2,000 different phenotypes, mostly different kinds of diseases. So all this data, it's not massive. We are not talking about petabytes or even terabytes. The sort of legacy format, how to store this kind of genetic data, it's actually a gzip compressed text file, and it's not efficient, of course. But then in compressed binary, it's currently less than a terabyte. And in our research, it's a genetic data that takes up the most space. It's not a massive amount, but compared to everything else, like the phenotype data here, That one is really, really small. So then finally, when we run analysis across this dataset, for example, we have to do more than 44 billion statistical tests, actually probably much more than that. And each of these tests will be done over all these individuals. So it requires some CPU cycles. And we run this in Google Cloud. We have a quota of 100,000 preemptible CPUs there. And we are actually currently doing this, and then every half a year we repeat our analysis with more data. And a year ago, Google said that these are actually the largest batch compute jobs that are being run on their compute engine. That's all I have, and thank you for your attention. Happy to take any questions. Yes, the question was if this is a Helsinki University project, and these are two separate things. This FinGen, a lot of this work is done in Helsinki, yes, but this is a collaboration of all Finnish universities and biobanks. And then quickly about the COVID, that's quite different because of the pandemic situation. It's just people who have time to invest on that will do it. Right, the question was if I do a lot of collaboration with private corporations. For example, in this FinGen study, this is largely funded by pharmaceutical companies. So that makes this a little bit different in the sense of how things are being done. So this is not a very academic project. So those companies actually expect us to do stuff on time and so on. But it doesn't. I'm quite happy with that kind of collaboration actually, because our aims in academia and in industry, they are not very different. Right, the question was if the companies that do genetics around Helsinki, if they are competition or collaboration. And actually, they indeed do a lot of these clinical things. They sequence people who might have a rare disease, and they find individual variants in a smaller set of people. So the research questions are a little bit different. And in the future, for sure, there's more room for collaboration as well, but it's not competing thing to most of what we do. But they shouldn't hire our PhD students anymore. Yeah.
Just out of curiosity, are you using PySpark or Spark to do anything? You didn't mention that in that slide.
Yeah, the question was if I'm using or we're using PySpark, and I have tried it a few years ago, but I'm not familiar with it, and I'm not using it. Well, I don't know if some people do use it, but we handle our big workloads differently. We don't. It's not necessary to use PySpark. Maybe it could be useful for some things, but yeah. Yeah, the question was where you can find these articles that I mentioned. Easiest, if you have a phone in your hand, you can Google the title. You can find it on Nature website, several places in the web. I can also show you after this. The question was how expensive is it to run this kind of batch compute job in the cloud? It depends quite a bit on the availability of the preemptible machines. Those are the kinds of machines that you can reserve from the cloud, but they might go down at any point depending on demand and mysterious things. But the cost is for one analysis round like this, it's tens of thousands of euros.
Are you using CPU cores or GPU cores?
Yeah.
Provide both?
Yeah, the question was whether we are using CPUs or GPUs, and this far only CPUs. Again, for some compute tasks, probably it would be good to use GPUs. If you have more insight, let's talk more.
Definitely, if you're using Python, the most used GPU core language is Python, I believe.
Okay, cool. I haven't done any GPU computing, so I don't. Yeah. Yeah, the comment was about the price difference between preemptibles and non-preemptibles. So, yeah, I can say at least in Google Cloud, the preemptibles cost 1/5 of the regular CPUs. But when they go down all the time, it's getting more expensive. But we are very happy with the preemptible CPUs actually in general. But it depends on, now it's going to be Black Friday soon, so we have to be fast with the current analysis because right, the question was about R and Python, which one is better, and if R is used in academia. I think it's a historical thing partly because R has been used among scientists for a long time. It was actually developed for that purpose, and it was developed for statistics. So for a long time, students have learned to use R if they haven't known any other programming language. I think much of the current use of R comes from that. I'm still not sure if R even has a syntax, but yes, the question was if I'm using or planning to use any machine learning. I know there have been some research in genetics using machine learning, but I have to say I'm not at all familiar with that stuff. So my work is this far just more like traditional statistics, but let's see in the future what comes out. Cool, thanks.
Okay. Thank you, Juha, and thanks everyone for coming. The event will continue, and I'm sure Jari will give you the details of how long we'll continue here. I just wanted to come here and advertise that we're having the next meetup at Volt, because Jerry was so miffed that he missed the sign up for this meetup that he had to come talk here, and then he had to agree to arrange the next one, so he won't have to be on at meetup.com to sign up for the next meetup either. But we'll be having the next meetup in the beginning of December at Volt. I expect to publish the event any day now. But in case, come on. Yeah, and if you're interested in talking either at Volt in December or possibly at Futurize in January, then let me know. We're always looking for speakers, and today's speakers were hard to beat, but we also do take mediocre presentations at this meetup. And thanks everyone for coming.